potential along this way will make the neurotransmitter to get released and excite the next neuron. This is third. But the movement of this action potential, like this action potential in this direction, will never release the neurotransmitter and will never make the first neuron to generate the action potential. That is the meaning of unidirectional. It means, suppose I am trying to draw the diagram again. This is the first order neuron, this is the second order neuron, and this was the axon hillock, and with green color I will mark the direction of action potential. Say this action potential is moving in this direction as well as in this direction. Once it reaches over here, it will release the neurotransmitter and make third neuron excitable, but it will never release the neurotransmitter and make this neuron will never get excited. So what is the use of moving the action potential in this direction? It is very significant. Why? See, this neuron, the second order neuron, receptors are there. It generated EPSP, excitatory postsynaptic potential. So there are the sodium channel 1 over here, here. So when the action potential is generated, few of the sodium channels which are lying along this segment, they are still opened, or we can say this segment is still in depolarized state. Okay, depolarized. If the second stimulus comes from the first order neuron, second stimulus comes, that will make this second order neuron again excitable. But for this second order neuron to become again excitable or again depolarized, it first has to come back to the resting state. From the depolarized, it has to be repolarized. So the direction of the action potential from axon hillock in opposite direction, it kind of flushes. It flushes off the opened sodium channels, means it makes this segment or brings it back to the resting state, repolarized, so that it's ready to get excited again by the second impulse. That is the significance of movement of the action potential in this direction. Okay, and then coming to the mixed synapse. Mixed synapse means a particular kind of the synapse where both chemical neurotransmitter are also getting released and there is a connection of the electrical one. All these like the mixed synapse, and they are not present within human being. Okay, so coming to the chemical, we have covered, electrics we have covered. Now the anatomical classification, that was the physiological classification, electrical, chemical and mixed one. Now in this one, anatomical classification, depending on which part of the nerve fiber is making connection with which part of the next neuron. If axon is making connection with the dendrite, then it is called as exodendritic. If axon is making connection with the body, soma, then exosomatic. Axon with axon, exoaxonic. Soma, body with dendrite, somatodendritic. Dendrite with dendrite, dendrodendritic or somatosomatic. Okay, likewise. So these are different diagrams. We will just quickly go through this one, quickly pass through. And instead of the chemical synapse, we have covered this. I won't go into detail. Just remember, like this is the synaptic cleft. This part is the synaptic cleft. This is presynaptic, and yes, this part, the postsynaptic membrane, in this one, it will have, particularly this part, this is called a synaptic density. It will have very high number of the receptor for the neurotransmitter. Okay. Now this one also we won't go into detail. Now see this one very quickly. Like there is a transport of the electrical impulse. Not the electrical impulse it's showing, like yes, the neurotransmitter, it is always synthesized in the soma, body part. It will get synthesized in the body part. It will travel, transport of the neurotransmitter within the vesicles will happen, and it will reach at the dendritic point. See this one, these neurotransmitters, they are getting transported at this end. When the action potential moves from this soma part along the axon, like this is the moment of the action potential. When the surface becomes depolarized, then outer side becomes more negative. So what will happen when this current reaches over here? It will open up the calcium channels, and these calcium channel, along with these calcium channels, calcium will move from outside environment to the inner environment, and this calcium will help in making the vesicles which has got the neurotransmitter within them to get attached to the surface, and exocytosis of these neurotransmitter will happen. Now these neurotransmitter, what is the fate of these neurotransmitter? These neurotransmitter will go and get attached to the receptor, and they will make these receptor activated, and ultimately they will get washed away. Washed away means either they will get broken down in their constituents, or they will be taken up by this one, reuptake of the molecules by the transport proteins. They will be taken up, and then it will be broken down into their constituent materials by MAO. MAO means monoamine oxidase. They will get broken down, and again they will get synthesized. So this cycle keep on happening. Okay. So we can say the fate of the neurotransmitter is enzymatic degradation, reuptake, or they might get diffused into the bloodstream, then get metabolized within the body, either through the liver or the kidney. They will get excreted out. Very little amount, but major chunk is they will get taken back. Reuptake, enzymatic degradation will happen, and then resynthesis of neurotransmitter. Okay. Now this part, the postsynaptic, like we have said, there is presynaptic neuron action potential generation and its movement along with the synaptic cleft to the postsynaptic neuron. Now what will happen to the postsynaptic neuron? Like, okay, over here I will use this slide. The presynaptic neuron might release the neurotransmitter which can be either excitatory or inhibitory. And what are the excitatory neurotransmitter? The excitatory are glutamate and aspartate. Okay. What about the inhibitory? Inhibitory are, these are like two examples each. There are so many others. GABA. Okay. Major inhibitory one. And the second one is glycine. Okay, very good handwriting, right? Okay, so depending on, say, the presynaptic neuron, it might be, we can say, this is one neuron, then there is a second neuron, then there is a third neuron. So this neuron might be releasing excitatory. I'll mark the excitatory neurotransmitter with the green one. Okay, suppose this one is excitatory, so it will release these neurotransmitters. Okay, now the second neuron might release, just take the example of inhibitory with the red one. Okay, so it depends. Just remember this thing. This neuron action potential happened over here. It released neurotransmitter, and those neurotransmitter happens to be the excitatory. If it is excitatory neurotransmitter, then those excitatory can be glutamate or aspartate. Then it will make the second order neuron to generate the action potential, and that action potential will be generated by generating excitatory postsynaptic potential, that is the EPSP. Now when this gets stimulated, it will release, because it happens to be inhibitory neuron, so it will release the neurotransmitter that happens to be inhibitory. Now what this inhibitory neurotransmitter will do to the next neuron, it will generate the potential, and that generated potential will be IPSP, inhibitory postsynaptic potential, because these neurotransmitter will be either GABA or glycine. So whether there is generation of EPSP or IPSP, first and foremost it depends upon which type of the neurotransmitter is getting released, the excitatory or inhibitory. There are so many other reasons, but this is the first and the foremost reason. The other one, like say, see, the excitatory postsynaptic neuron, depolarization of the postsynaptic membrane by excitatory neurotransmitter, that can be the glutamate or the aspartate. How does it generate the EPSP? It will bind with a specific receptor protein and opens the ligand-gated sodium or calcium channels. Whether there is opening of the sodium or the calcium, it means there will be movement of a positively charged ion into the neuron. When there is movement of the positively charged ion into the neuron, then obviously the outside will become negative. That is called as depolarization. Since a very small area of the postsynaptic membrane develops increased sodium permeability, only a brief polarization will be followed by slow decline to resting membrane potential. What does it mean? It means like there is generation of a very brief polarization, Suppose this much polarization happen. This is a resting line, resting membrane potential. The sodium channel open up, there is generation of this much potential. Then again it falls back to the resting membrane. So this part is called as excitatory postsynaptic potential. This is not propagated. It won't transmit over the cell. So it can only depolarize the adjacent membrane, the surrounding membrane, due to the local currents set up passively. So it's a gradient response. It is not propagated. Because it is not getting propagated, it is a gradient response. It won't follow all or none law like the action potential. And it shows the summation. This one we have covered so many times. Two type of the summation. Again, we will talk about these two. Temporal as well as the spatial. What is temporal? Temporal is timing. Means, suppose this is one neuron and this is the second neuron, that is the postsynaptic neuron. We stimulate it, this one time. It will release the neurotransmitter, then make this thing to excite. Then there comes the second impulse of the same neuron. It will again, like after very brief interval, we are giving very much less time, okay, like 3 or 4 consecutively. Then whatever the EPSP is happening from the first stimulus, second, third and fourth, they will get added up, summed up. That is called as temporal summation. And what is spatial summation? Spatial summation is, I'll use this color. Suppose this is the postsynaptic neuron. Okay. Now, this postsynaptic neuron is having connection not only with one presynaptic neuron. This is one presynaptic neuron. This is the second presynaptic neuron. This is the third one, and this is the fourth one. If we collectively stimulate all these 1, 2, 3, 4 presynaptic neurons, all 4 of them will release their neurotransmitters, okay, and this postsynaptic neuron will get excited to the level by the sum of all these 4 presynaptic neurons. Suppose it is generating this much EPSP, this one is generating this much, third one is generating this much from the baseline, fourth one is this much. So this is the baseline. These 4 will totally get added up and will generate very high amplitude. That is the spatial, means space at different space interval. That is the spatial. So this one will explain it by this thing. Okay, diagram. See, actually all this slide we have covered up. So that's why I am going quickly. See, this is repeated stimulation of one excitatory nerve. Like there was one presynaptic neuron and then there was a second, that is the postsynaptic neuron. So only one, we are talking about only one neuron. Repeated stimulation of this excitatory nerve. So initially there is generation of EPSP of this much. Then by the second, third, fourth, they collectively it will get added up, and at one stage it will cross this line, and this line is the threshold. Like the resting membrane potential of the neuron is minus 70 millivolt. If you are taking, if there is only one stimulation, it will make it either to 72 or 73. And suppose this one threshold we are talking over here is minus, say minus 50 we are talking. So if we are stimulating this now with one impulse, and that one impulse is generating the EPSP of 5 millivolt, then it will make from minus 70 to minus 65, but it will not reach to minus 50, that is the threshold. But if we stimulate repeatedly 4 times, then 4 plus 5, that will be 20, and it will cross the minus 50. 70 minus 20 will be minus 50, minus 70 minus, it will become cross the threshold. As soon as it crosses the threshold, there will be generation of the action potential. That is how the this one temporal summation happens. Okay, now in this example, the second example shows, this is the example of spatial summation. Means EPSP is produced due to stimulus at single synapse, and then these are the EPSP produced at multiple synapses. Like say the postsynaptic neuron is only one, but presynaptic neuron there are multiple, 1, 2, 3. Okay, so this, all these 1, 2 and 3, they get added up and makes it to cross the threshold. Okay, so that is how the EPSP is getting some. So similarly, just the difference between the EPSP and IPSP will be like in EPSP, the neurotransmitter released was glutamate and aspartate, but in the case of the IPSP, it is glycine and GABA. So what happens over here in the IPSP is the inhibiting neurotransmitter, it will cause the opening of either the potassium or the chloride channel. What was happening in the EPSP, excitatory? It was making either the sodium channel or the calcium channel opening, and whether the sodium or the calcium, whatever, like whether the sodium is getting opened up or the calcium is getting opened up, the positive ion was moving inside. But in this scenario, if the potassium channel gets opened up, where is the maximum concentration of the potassium? Inside the cell. So obviously there will be a rush of the positively charged ion, positively charged potassium ion from the cell into the outside environment. It means the outside will become more positive. If there is opening of chloride channel, what will happen? The concentration of the chloride is more on the outside, it will move inside. So in either condition, whether there is opening of the potassium channel or opening of the chloride channel, the inner will become more and more negative, so leading to diffusion of large number of potassium ions from the neuron to the ECF or the chloride to the interior. So ultimately, inner membrane of the neuron will become more negative. It means we can say it won't get depolarized. It will become more negative or hyperpolarized. That is called as inhibitory postsynaptic potential. And similar to the EPSP, there is summation, either the spatial or the temporal. Okay, and it ultimately leads to postsynaptic inhibition. So development of this one we have covered. There are only 3 steps. One is the synaptic integration, generation of the initial segment spike, and